Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Long. We're going to be doing this video during this coronavirus shutdown. I'm sure all of y'all are tired of this disclaimer, but these lectures are being done rather quickly and crudely, and they're really targeted towards students taking my Human Anatomy and Physiology class. I hope to one day be able to do slightly better videos, if not much better, with nice editing and very well done, much larger board, better format. Nonetheless, I have what I have to work with, and so we're going to knock these out. This lecture is intended for my Human Anatomy and Physiology 2 course, Biology 2402. We just finished the female reproductive system, so this video is going to be on development. We're going to start with a meiosis and what's called gametogenesis. We're going to go through fertilization, implantation, and all of the processes. There's so much information here that usually embryology is taught as an entirely separate course from human anatomy and physiology. But when you're going into the health sciences, there's some of this information you need to know. There's no way I could encompass everything that's necessary for you to know, so I'm going to hit some main highlights. Okay, I'm going to start in my note set on page 102, I believe it is. We're going to start talking about meiosis and gametogenesis. In order to do that, I want to give you a little setup because a lot of students may not either have had the biology or remember this. But, you know, we have this double helical molecule called DNA in our cells. And DNA has all these bases that are bound to each other with some hydrogen bonds into double helix coiled structure. There are some little proteins called histones that can bind to the DNA, and the DNA actually starts to wrap around it. And as we bind it around the histones and wrap it, and then have another histone and wrap it, what we end up with is a really dense body of DNA, a really dense structure. And we end up with two pieces that are connected by a little protein in the middle. And these pieces out here are each called a telomere, telo meaning end. So I have two telomeres. And there's a protein located in the center called the centromere. Okay. The telomeres are actually the coiled up DNA along the histones, very tightly condensed, okay? And we just draw them as these little loops, but really there's a whole bunch of long piece of DNA all coiled up, almost as if you took a long cable and you wrapped it around your fist until it was about this big. Instead of being 12 feet long, it might end up in a little ball this big. Well, that's what this is. The whole structure is called a chromosome. Okay, this would be a chromatid when the DNA is, it's called chromatin, when the DNA is all uncoiled and really long. But when we coil it up into a dense body, we call that a chromosome. Now, this chromosome itself consists of what we call one single chromatid. So there's some confusing terminology here, but a chromatid is this entire piece. At one stage in its life cycle, in the cell's life cycle, every chromosome is made up of a single chromatid that has two telomeres connected at a centrally located centromere. Now, as you know, during mitosis, when we undergo cell division, we actually have to make a second copy of this chromosome. We're literally going to copy it. And when we coil it up, in order to keep track of the two copies of that one particular chromosome, a lot of times we can stick the two chromatids to the same centromere. So now I have a chromosome that has two chromatids. It's got 4 telomeres. Okay. So when we're not undergoing mitosis, chromosomes are uncoiled as chromatin, and we're reading the DNA and all of that. We can condense the chromatin down into a chromosome that consists of a single chromatid with 2 telomeres and a centromere. During cell replication, during mitosis, the second copy of the chromosome will also condense down into a chromatid with the 2 telomeres and a centromere, but the centromeres get stuck together, so it looks like this funky little thing. And so we tend to draw chromosomes looking like little X's, like this. Okay? That's when they're undergoing mitosis, and we freeze the picture. I wanted to give you some of the anatomy because of a chromosome, because we're going to be going through 2 phases, mitosis and meiosis, and I'm going to be using this terminology. Okay, so now let me erase all of this. This is where the notes begin for you guys. Okay, now our cells divide by a process called mitosis. Most of our cells do. And so the cells that divide by mitosis are called somatic cells. Somatic cells are all of the cells of the human body except for what we call the gametes. When we talk about the gametes, another way to say gametes is the sex cells. The cells that you make depending on what your sex or gender is, right? So the gametes can be called the oocyte if you're a female, or they could be called the spermatocyte or spermatozoan if you're a male. Those cells undergo meiosis. So somatic cells divide by mitosis. The gametes, the oocyte and the spermatozoan, divide by, a specialized process called meiosis, or some people say meiosis. Now, we know the phases of meiosis. The cells are living in interphase. I'll put a little I here for interphase. If I were to draw a cell in interphase, and let's say I have a cell that has only 3 chromosomes. Our cells have 23 chromosomes, and then there's a second pair, so there's 23 pair of chromosomes. Here's what I'm going to mean, okay? So let's say I have a chromosome in here, and this is chromosome number 1 from Dad. I'll color it in a solid color. Well, Mom is also going to give you a copy of chromosome number 1. So there's one from Mom. Now let's say this is chromosome number 2 from Dad. I'll leave it hollow. And this is the chromosome number 2 from Mom. They're not going to always be next to each other. They're in the cell moving around. And I've drawn them condensed down. And let's say that this one that has stripes on it is going to be chromosome number 3. So we're tracking 3 pairs of chromosomes. You get a set from Mom and a set from Dad. Now, during interphase, if the cell is going to divide, is when the cells are all going to replicate that chromosome and condense it down. And by the time we get to prophase, what we have inside of our cell, and I've broken down the nucleus already, although the nucleus would be around all that, but these chromosomes are all going to replicate. For example, if this is chromosome number 1 from Dad, it's going to have its second copy here like this, stuck with their centromere. It's got 2 chromatids, 4 telomeres. Now, here's chromosome number 1 from Mom, the original, and here's the copy stuck to it so we can keep track of it. If I did chromosome number 2 from Dad, it would have a second copy. And the same thing for chromosome number 2 from Mom. And if I wanted to do chromosome number 3, I guess I could. I would get this. And the chromosomes are going to condense into these dense little bodies. Now this is where it gets interesting for us. By the time we get to meiosis, we're going to see a big difference here. When I get to metaphase, the chromosomes line up in a very unique fashion. So if I did chromosome number 1 from Dad with its 2 copies, and chromosome number 2 from Dad, that's 2 copies, and down here I'll do chromosome number 3 from Dad with its copies. Mom's chromosomes are going to line up very similarly. Chromosome number 1 from Mom, number 2 from Mom, and number 3 from Mom. They'll all be lined up in here. In anaphase is when we separate those chromosomes. And what you literally see is the centromeres will break them off at the telomere. The spindle fibers will attach to the centromeres and break them apart and pull an entire copy of chromosome number 1 from Dad in one direction and another copy in the other. Chromosome number 2 from Dad and number 3 from Dad. So you can see how I started with a single chromatid. I replicated it, and now I stuck the 2 identical chromatids together into a chromosome. But then I line the chromosomes up, and I separate them so that I get chromosome number 1 from Mom, 2 from Mom, and 3 from Mom. And then in telophase, as you guys know, I'm not going to draw a whole other picture because I'm out of space, the cell would divide into 2. And if I look at it, This cell has chromosome number 1 from dad, 1 from mom, 2 from dad, 2 from mom, 3 from dad, 3 from mom. Just like this. And the two daughter cells end up with a full complement of genetic material. Because I have 2 copies of everything, this cell is said to be diploid. Di meaning 2, and ploidy meaning the number of chromosomes. I have 23 pairs of chromosomes, or in this drawing I have 3 pairs. Each daughter cell is still going to be diploid at the end of mitosis. Now I didn't draw out all of telophase, but you get the 5 phases. Now when it comes to gametogenesis, gametogenesis means to make the gametes. There are 2 subtypes of gametogenesis. One is called oogenesis, that's the production of the oocyte. The other one can be called spermatogenesis, the production of the sperm cell. There are 2 subtypes of gametogenesis. Gamete means the seeds, and so for females the seed is the oocyte and the spermatozoon for males, and they undergo oogenesis or spermatogenesis. So when it comes to gametogenesis, the cell divisions occur by meiosis. Now I need a little bit more room here, so I'm going to erase some of this. Meiosis goes in 2 rounds. Now if I were looking at let's say a spermatogonium or the oogonium, the cell that's eventually going to divide by mitosis, but then produce that first cell that's going to start the meiotic divisions. So if I'm going to start in meiosis 1, in meiosis 1, we're going to have interphase, prophase, metaphase, anaphase, and telophase. We're going to have the same phases. Interphase would look very similar. Prophase would also look similar in the sense that I have all my chromosomes. I've got chromosome number 1 from dad, chromosome number 1 from mom. 2 from dad, 2 from mom, 3 from dad, 3 from mom. When we get to metaphase, something different happens. If I were to draw the cells in meiosis 1 in metaphase, the way the cells line up is not, or the chromosomes line up is not the same as it is in mitosis. So if I have chromosome number 1 from dad here, and I think I made that one a solid chromosome, and I got its little chromatids, the 2 sister chromatids, right? Well, it turns out that chromosome number 1 from mom is going to bind to the centromere for chromosome number 1 of dad, and we actually end up with 4 chromatids equals 1 chromosome. They call that tetrad formation, because tetra means 4, right? So in tetrad formation, we have the 4 chromatids on 1 chromosome. Chromosome number 2 from dad might look like this, and chromosome number 2 from mom is going to stick to it. And chromosome number 3 from dad might look like this, with its little stripedness. And chromosome number 3 from mom, you can see how from this lineup, I literally break each chromosome in half, but in this lineup, I'm going to pull an entire tetrad across, okay? Or an entire chromosome with 2 homologous chromatids on it. So when the centromeres attach, they're literally going to pull both of dad's chromosomes in one way. Now it's still chromosome number 1, but it's both copies from dad. And chromosome number 1 with both copies from mom are going to get pulled in another direction. So when I get to anaphase 2, or anaphase 1 I should say, the cell will start to look like this. I'm going to pull all of this in one direction, and I might pull number 2 from dad in one direction and number 3 from dad in this direction. And therefore, mom's chromosomes, see if this marker writes a little bit better. Here's number 1 from mom, both copies attached to their centromere. Here's number 2 from mom over here. And here's number 3 from mom over here. This gives us some genetic variation because now chromosome number 2 in this person comes from mom, but chromosome number 1 from this person came from dad. So you got a little bit of both mom and dad's DNA. And because, if I really look at it, here, I have 2 copies of chromosome number 1, one from mom, one from dad. When this splits, I'm going to have 2 copies of chromosome number 1, but they're both going to be from dad. So I don't really have that second copy. I don't have that paired chromosome. That's different. Okay? So we call this, in anaphase 1, this is called the reduction division. The reason we call it that is because we started off with a diploid cell. By the time we get to telophase here, the cell is said to be haploid. So when I draw this 2 daughter cells, when this thing splits, one cell is going to have these chromosomes. This is getting a little bit hairy, but anyway. We're going to have this and this. But look, I'm missing all of chromosome number 1 from mom over here. I'm missing all of chromosome number 2 from dad. And the opposite cell would have the exact opposite over there. I'm not going to draw it, but you get the idea. We have divided from a diploid cell, a cell that has 2 sets of chromosomes, into a cell that has 1 set of chromosomes. Okay? So it's important to note that in metaphase of meiosis 1, we get the tetrad formation. And in anaphase of meiosis 1, we get the reduction division. Now, I hope you could follow all that. It's always no fun teaching this. One of the other things that, by the way, can happen is that we can actually swap pieces of chromosome between 2 cells in a thing that's called crossing over. And these 2 chromosomes can exchange some information, leading to even further genetic variation. That genetic variation is important for survival. If there's a disease that attacks a population and they are all genetically identical, if it can kill one, it will kill the other. If you have a group of cells that are not identical, or human beings that have different genetic makeups, then maybe some of them have a whole new novel antibody that might be able to fight off a specific infection. They might be able to produce the antibodies to fight off an infection where some people were dying, but this person has enough genetic variation in their immune system that they might be able to withstand and survive, so they live through something like the terrible pandemic that we're undergoing. Anyway, now, the other crazy thing that happens is we go through meiosis 2. In meiosis 2, we skip interphase. We go right back to prophase, metaphase, anaphase, and telophase, and it looks a lot like this. And essentially, I can go from 1 diploid cell to 4 haploid cells. The spermatozoa divide equally and produce 4 spermatids. The oocyte does not divide equally when it does these divisions in meiosis. All of the cytoplasm and organelles go to 1 cell. The other cell just ends up being a little membrane with some DNA in it called a polar body. And so we don't get 2 functional cells out of meiosis 1, usually. And then when we go through meiosis 2, this cell will divide again and kick out another little membrane with some DNA in it, but all the cytoplasm and organelles stay in 1 particular cell. And so you get a really large oocyte and a couple of little polar bodies that are just extra DNA. So all of this information is explained to you verbally on page 102. There's some more information on page 103 that goes through all of this and talks about its importance. Now, I'm also going to have another video up that just explains meiosis, but it's going to be a video that someone else does, and it's so good that I can't replicate it. It's just good enough that you can see it all happen if you want to watch that. Now, a couple of little details that I want to explain, and then I'm going to end this video, and we're going to have to do one more video for development, or else this one will end up being super long. And the first video on the male was 40 minutes. Okay. Now, when it comes to gametogenesis and spermatogenesis, or when it comes to all of this stuff, by the time we get to the end of the process, we have 23 chromosomes in each cell. Man, I need to get a different red marker. That one's hard to erase. So, look, Pardon my back, but when it comes to the chromosomes, there's a period of time, especially when they do what's called an amniocentesis. They can stick a needle through the umbilical cord and pull out some amniotic fluid. And in that amniotic fluid, there will be some cells floating around where the cells were dividing, and there may be extra cells in the tissue that will be lost during birth. When they catch those cells, there's a point in time where the cells were all replicating their DNA, and so all of the chromosomes have 2 chromatids, and they've numbered them and put them in a certain order. The way that they number them is by their size. The largest chromosome became chromosome number 1, and also you can pair it up with its other copy by the banding patterns. But this would be chromosome number 1 from dad and its identical copy. This might be chromosome number 1 from mom and its identical copy. The next chromosome, chromosome number 2, is going to be slightly smaller and will have a different banding pattern. They put these stains on it, and you can see these dark bands on them. When we go to chromosome 3, it gets smaller, and the banding patterns change. By the time you get to the 23rd pair, you'll get a chromosome from mom and a chromosome from dad. They'll be the smallest ones. If they both look like this, which we describe as being XX, the offspring is going to be female by gender. If the chromosome from dad, the 23rd one, is missing one of the telomeres, but the chromosome from mom is not, then the baby is said to be XY. I drew it backwards, but if you're XX, you're going to be female. If you're XY, that missing piece of genetic information makes you entirely male by gender. So, I know we can get into gender identification and all that other stuff, but physiologically speaking, you're usually either male or female. There are some people who are hermaphroditic, and there's some confusion amongst the genetics and the biochemistry, and they can develop a rudimentary vagina and a penis. Usually neither is functional. And so hermaphrodites exist. But outside of that, when it comes to our physiology, we usually think of someone as male or female. In this whole age of how you identify your gender, you might have a male that identifies as female. Physiologically speaking, no matter how much chemistry and how much hormones and everything we pour into you, and surgeries and everything, genetically your body built testes and is trying to produce testosterone like crazy, and therefore it's important that we keep this in mind when we're treating patients. Males tend to have higher incidence of certain cancers than females do, and vice versa, and other diseases like heart disease and high blood pressure and other things. There are certain diseases and disorders that are more abundant in one gender or the other, and it's important to kind of know that information. By no means am I telling anyone how to think or feel or believe about any of this stuff, but when it comes to the biochemistry of the body and pharmacology and other things, it is important for us to know what the genetics are of the cells, which has a huge change in the biochemistry of your body. One of the things we talked about earlier in the semester when we did the endocrine system is the secondary sex characteristics. So for males, that's the body hair pattern. It's the fat distribution on the body, the musculoskeletal development, the deepening of the voice, and all these other things that happen, the falling out of the hair, and all these male gender-specific attributes. We call those the secondary sex characteristics. In females, the secondary sex characteristics would be similar things: the fat distribution on the body, the changes in hair and growth and development, breast tissue, and the genitalia. The primary sex characteristics are you male, do you have a penis, or are you female, do you have a vagina? The secondary sex characteristics are all the things that happen at puberty that make you more sort of male-looking or female-looking. Those secondary sex characteristics also come with changes in the brain. There are differences between the male and the female brain, and a lot of psychology and neurobiology based upon that. So I think it's important that we still kind of keep an open mind to this idea that people are either male or female by birth, And then if you want to identify as any other gender, that's between you and your family and your physician and anybody else. I don't want to judge any of that. I don't want to get into that. But physiologically and eventually medically speaking, the knowledge of this information is going to be important for properly treating people for certain things and testing for certain things like cancers and diseases and other things. Anyway, I'm going to wrap it up there. I don't mean to get too philosophical. I certainly don't want to get political because I do believe strongly in education. I do not believe in indoctrination and telling students what they must believe. You can disagree with me all you want, and that's fine. Anyway, we got one more video coming, folks, so I hope you learned something. I hope you had as much fun as I did, and I hope to see you on the flip side in the next video. See you.